By the way, would you mind to share, for example, end-to-end life cycle to find a new drug in your industry? Because our audience usually from the software engineer or the tech lead in the tech, I mean the high tech industry. Yes. Would you mind to share some more context in your area? Yeah.
Sure, I'm happy to. So, drug development, as I mentioned, could be 10, 15 years long from a discovery of a target to actually getting a treatment to patients. And that whole process involves a lot of it is the research at the very beginning that we're seeing being done in the academic labs, as well as larger companies that may have the resources to really explore the biology. We've come a long way in terms of what we understand, and we have new technologies like CRISPR and all the omics type of studies that gives us insights. But like you said, we're still very much it's a slow process to identify new targets. But with AI, hopefully we can have more hypotheses of what are relevant targets in a disease. And once you identify those, then you're testing them in usually cell culture models where you might remove the protein, target from the cell, and then you see how does the cell behave. And that gives you an idea of whether it's actually relevant, in the biology of the cell in the context of whatever the disease you're looking at. So in my case, we're looking at neurodegeneration. So, a lot of our targets are looking at the immune cells in the brain. And we look for targets that can make the immune cells be better at fighting off the neurodegenerative disease processes. And so once we identify those targets, then we develop molecules that are going to target those proteins that usually are protein targets to either block them or to enhance the protein. So there are different ways you can kind of change the way the biology works. And these proteins, in biotechnology, you're generating antibody proteins as the drug. There are also drugs called small molecules, which is based on chemistry to go and be able to change the biology. But the difference between those two is that the large molecules, because they're antibodies, they're very specific to the target, which means that the off-target effects, side effects, are lower than if you have the small molecule. So biotechnology was born to really generate new treatments that are basically antibody-based, and these are the large molecules, we call them. And so I'm going to talk about that type of medicine. So once you have those molecules generate, usually you make different versions, and then you test which one is going to be the best one to go after your target, because you need to see how long does it stay in the system, is it actually hitting your target. And that's when you start going into what we call translational sciences, where we're looking for ways to better understand the actual drug, the antibody in this case, in an organism. And so that's where we can test it in the mouse, or also in non-human primates, monkeys, to be able to see how long does this drug stay in our system? Is it hitting the target that we intended to hit? Are there any other safety issues that we see when the animal is taking this drug? So safety and tolerability is the very key thing before the FDA in the US at least will then grant you, the permission to be able to test in humans. And so if you do get approval to move into the clinical trials, the first phase of the trial is really to assess the safety and tolerability of the drug. And usually you go at different doses to find the right titration of the drug. And then once you have established safety, then you go into the population with your disease. So in our case, if we're looking at dementia patients, we will try to find the dementia patients to enroll. Then we will give them the drug in the dose that we find to be safe, first of all, and then hopefully efficacious, that's able to, be effective for them. And in the phase 2 study, if you see that there is safety in your patient population, then you can go into a large phase 3 study, which we call a pivotal trial. And that's where you are often testing with people who are on treatment on your drug and people who are not on your drug on placebo, so that you can assess, is your treatment actually working in the patient population on a large scale where you can do statistical analysis. In areas, like mine, where in treatments of the brain, sometimes it's hard to really assess like cognitive decline. It's a little more variable when you assess this. So we also leverage things called biomarkers, which are often proteins in the blood that you can measure. And that way, these proteins are basically what we might think represent different aspects of the disease. And if you can see that if your patient under treatment has a change in these proteins, that might indicate your treatment's also working. But yeah, that whole process from discovery to clinical trials, depending on the disease, could take quite a long time. And often the long time is, the discovery piece, because so many of the targets you identify are not good targets, or they end up having drugs that really aren't effective. And a lot of the failure actually can happen, before you even get into the clinical trial. So you have to discover and make a lot of bets before you find one drug that can then move into patients. And then, of course, in the clinical trials, that also has a high bar for what makes it through. So that's where if we can get more candidates in the funnel, that's when we can then have, the probability of having an actual successful treatment available at the end of the process. So hopefully that helps.
Yes, thank you. Yeah, thank you very much. Lots of, this kind of knowledge, and thank you for sharing the whole context about how to find and productionize a real, the human use this kind of build a drug. We have to the potential, the target, this kind of structure, and then lots of human in the loop, the scientists and the engineering to verify the safety and to phase 1, 2, 3, this kind of clinical trials. And we also need to double check the side effect. And finally, we will get the approval from the FDA. And everything actually looks like it's just about 20 years yet, because of patent. The protection, in the USA. And after that, I think the other countries, they will leverage. I know some stories about the India, actually, after this kind of 20 years, the safe coverage for the patent, they will, localization.
Yes.
Yes. So it looks like, for the bioscience, this kind of field, the future is definitely pretty good, but for even current now, leverage this kind of generative AI, there are still lots of human in the loop, lots of human efforts to make it happen. So